It's question 2 from paper 1 of the AS 2017 paper. There isn't that much more demand from a GCSE question in this question. However, still only 7.6% of students managed to score the 2 marks for the first part of the question. Let's read through. It says a student investigated the effect of 3 types of disinfectant on the growth of lactobacillus bacteria. During the investigation, the student boiled the agar before pouring the agar plates, transferred 0.5 cubic centimetres of a diluted liquid culture of the lactobacillus onto each agar plate, left some agar plates as controls, added to the other agar plates different concentrations of the disinfectants as shown in table 1 on page 5. After 2 days, she counted the number of colonies of bacteria on each agar plate. So fairly simple so far. Basically, they've not given us all the steps, they've not given us all the detail, but they've given us the detail that we need to know. So, as mentioned before, I'd annotate the question as you read through it. I'd underline key points, key phrases, just so that your eye can flick back to key areas of the question much quicker than reading the whole thing through once again. And although this is true, it gives us that information in the stem of the question. So clearly the reason why we boil the agar after making it up is so that we can kill any bacteria in there. So we prevent contamination, so that we know that the results that we get are the results. So in answer to this question, you could have written, so there's no bacteria present, or to avoid contamination. The second part of the question asks us to explain the purpose of transferring the same volume of liquid culture onto each agar plate. The next part of the question is where it went very wrong for a lot of students. Students didn't seem to know what liquid culture was. So just to be clear, agar that we see in the agar plate is nutrient agar. It's got all the ions and minerals that bacteria need, plus the seaweedy bit, the agar bit, to make it go into a jelly. Liquid culture is just the nutrients without the jelly. You would make up the liquid culture when you want to grow a certain concentration of bacteria. You would use the recipe that would be given to you, and it would just be a case of adding the correct nutrients and ions and minerals and inoculating it with a small amount of bacteria, and then you would leave it in a McCartney bottle to grow. The liquid culture may appear cloudy, and again that's because it's got ions and minerals dissolved in there, but also if the concentration of bacteria has got really high, it may just appear cloudy due to that. So in answer to the question of why did we transfer the same volume of liquid culture onto each agar plate, it's so that the same number of bacteria are transferred to allow comparisons afterwards. For the next question, only 15.6% of students managed to get both marks for this question, which is crazy. It says the 3 disinfectants used by the student were Lysol, propantuol and ammonia. And there we have the table with the information in it. So basically, they put in 300 colonies of bacteria at the beginning, and then they increased the concentration on different agar plates, and after a concentration of 20 arbitrary units, they could see that it was 0, 0 and 240. So what does the actual question ask? Well, it says the liquid culture that the student transferred was diluted by 1 in 10,000. Use the information in this question to calculate how many bacteria were present in 1 cubic centimetres of undiluted liquid culture. So if we've moved the decimal point by 10 to the minus 4 when we diluted it, then we need to move the decimal point to 10 to the minus 4 before it was diluted. Also, the key to the question, and this is why I tell you to go to the question and to sort of scribble on the main points as you read through it, it tells us that during the investigation, the student transferred 0.5 cubic centimetres. So if 0.5 cubic centimetres was 300 colonies, then 1 cubic centimetre must have been 600 colonies. With that information then, all I need to do is times it by 10 to the 4, because I'm taking my dilution the other way, before dilution, if you like. So therefore, my answer, if I multiply them out, is 6 times 10 to the 6. And I write my answer in the expected standard form. Moving on to the last part of the question, it says the student concluded that the minimum concentration of propantuol needed to stop the growth of lactobacillus was 15 units. This conclusion is incorrect. Suggest how you could obtain a more accurate estimate of the minimum concentration of propan-2-ol needed to stop the growth of this species of bacterium. When we talk about accuracy, we're talking about how close we are to the true value, the real figure. Looking back at the table, we can see a drop from 195 to 0. That drop took place somewhere between 10 and 15. So we can't make the conclusion that it happened at 15. It could be 11, 12, 13, 14, 15, or somewhere in between those numbers. So to answer the question, we're going to say that we would investigate the concentration of disinfectant values between 10 and 15. And then if we found an answer between, say, 11 and 12 or 13 and 14, we would repeat the values in between those. I hope you found the video useful. Please do leave me a comment.